All right. Well, thank you all so much for joining today. My name is Bill, and today I'd like to talk to you about the very recent GENCODE upgrade that happened at GDC to version 36. I know we're doing this in the middle of AACR, so we'll try to get this recording out as soon as possible, so that people who couldn't attend today can watch it at a future date. So just to go over the agenda a little bit, the first thing we're going to do is give a short overview of the GENCODE 36 update. Um, Next, we'll go over a few in-depth changes to the GDC data and pipelines. This will be done by experimental strategy, and it's not going to be a section. This is kind of going to be sprinkled throughout, but one of the main changes here is synchronization of the TCGA pipelines to the other projects. So you'll learn a little bit more about that. Next, we'll talk about how to download V36 data, and then we're going to talk a little bit about how the update um, happened. And finally, we'll have a Q&A session at the end. During the presentation, feel free to type any question you have into the Q&A panel. There is a chat panel too, but please put your questions in the Q&A just to make sure that we do see them. So those will be answered at the end. So starting out, when the GDC first launched in 2016, files that use the gene model, and I say this because not all files do use the gene model, were generated using GENCODE v22. And recently, as of data release 32, this was just a couple weeks ago, files that use the gene model version 22 have been replaced with files that were generated using GENCODE version 36. So many files in the GDC have been replaced if you've seen it lately. So just to show a comparison of GENCODE version 22 and v36, you can see the gene count here is fairly similar. There are fewer duplicate gene names in GENCODE v36. And when you look at the shared genes across them, by gene names, um, around 60% are shared by gene name. Around the same amount are shared by gene ID when you include the version. So the exact same version is shared for about 32,000 of the genes. And then when you look at the gene ID without the version, so just without the decimal at the end, you get around 90%, um, or sorry, over 90% of the genes shared between GENCODE v22 and v36. So in terms of just the genes that are included, it is most of them. So that was just a basic overview of what the GENCODE upgrade was. But now I want to get into specifically what happened with the GDC data and the pipelines and how they've been processed either differently or similarly in some cases. So here is the RNA-Seq pipeline for GDC, a very simplified diagram of it. And as you'll see, we have three RNA alignments that come from STAR. STAR is used to produce expression quantification, which includes the counts and the transcript fusion files. HTC, don't worry about that for now. And for the structural variation data, we use Ariba and STAR, and we use that to produce transcript fusion files. So for the GENCODE version 36, in short, everything RNA-seq has been modified to a v36 version, a GENCODE v36 version. So that includes the alignments, the gene expression files, the splice junction files, the transcript fusion files. So basically, in short, all files have been changed for all projects in the GDC with RNA-seq. So a few more changes about the RNA-seq data that don't necessarily apply to GENCODE, but have been done in this release, and you should know about it. So the TCGA RNA-Seq in the past was processed using an older pipeline, and now TCGA RNA-Seq has been reprocessed the same as the rest of the projects um, with a more up-to-date pipeline. This includes now TCGA data, when it didn't before, has 3 alignments, a star counts file, and the fusion files um, from Ariba. So to replace the normalization methods that were formerly available in HT-seq, and sorry, I think I forgot to mention that before, but if HT-seq files were removed during the GENCODE upgrade, so we no longer support those files. Because if you remember, HT-seq had some additional files. They had HT-seq counts, HT-seq FPKM, and HT-seq FPKM UQ. Because those are now gone, a series of normalization methods were added to the star counts file to replace them. So I have kind of a snippet from an open access file that is available in the portal right now. And you'll see that at the very end here, we have the TPM unstranded, the FPKM unstranded, and the FPKM UQ unstranded. And the reason we have this is that the reason they're unstranded is because not all samples in the GDC were either stranded or unstranded, so everything is processed unstranded. And samples, if you have them, if a sample's in the stranded first category or stranded second, they can't be compared between them. So everything is processed unstranded, but we do include the stranded counts just in case you're able to, you have a group of samples that are included in one of these. And I do want to mention that a way to determine if if a if a sample is stranded, if it was produced um, using a stranded library, the in the n ambiguous row here, I think it's the 6th row down right here, they should have a significantly higher number of reads in the stranded first than the stranded second, or vice versa. And that will tell you that it is likely a stranded library. Moving on to whole exome sequencing and targeted sequencing. Um, if just to go over the pipeline, we start out with some aligned reads in a BAM file, usually tumor and normal. Those are combined into, those are both compared using several pipelines, and variant calling is used to generate some VCFs. The VCFs are then annotated and in some cases aggregated into collar level maps and then aggregated into ensemble maps that include all of the collars. So the changes that were done for GENCODE here is that the annotated semantic mutation, the annotated VCFs have been replaced with the v36 version. The aggregated semantic mutations, which are the ensemble maps, the controlled ensemble maps were also replaced, as well as the open access ensemble maps that are also called masked semantic mutations. The files that didn't change were the alignments and the simple semantic mutations, which were the raw VCFs. This change also includes the liftover data that was produced for the Foundation Medicine Adult Dataset project. So that went through these same changes where applicable. Another change, and you're going to see this is a pattern you'll see with TCGA for this release. TCGA whole exome sequencing was reprocessed using the same pipeline as other newer projects. So the TCGA alignments and the simple semantic mutations, the raw VCFs, have been replaced in addition to all those other files that we showed in the previous slide. In addition to this, you may recall that the TCGA maps were project level while all the other projects had aliquot level maps. And this has now been changed as of data release 32 so that TCGA v36 maps are now aliquot level and match with the rest of the GDC projects. I also want to go over some of the changes that happened in the exploration portal. So all of the genes and the mutations that you see in the portal exploration page have been regenerated using the v36 maps. As you probably know, this data is generated using the open access ensemble maps. And now, because those were replaced, the data in the exploration portal has also been replaced. So, in addition to this, there's a few other changes that you should know about. One is that somatic sniper data no longer appears in the exploration page. If you look down at the variant colors that are available, you will no longer see somatic sniper. Although right now the somatic sniper files are still available in the portal. The TCGA exploration data has been specifically changed, and I'm going to go over that in the next slide. So for TCGA, one thing about TCGA is that it was one of the first programs that the GDC had imported. TCGA mutations were processed in a special way because they had project level variant color maps. Now the TCGA has the same type of maps and are processed with the same pipelines that all the other pipelines are processed. The mutations on the exploration page are following suit. Some of these changes include now TCGA mutations require at least 2 variant colors to support them. So for each mutation you see, those are supported by at least 2 variant colors. There previously was a TCGA specific variant rescue step that is no longer supported. And mutations that fall outside of the target capture kit are removed in the masking step. So those will no longer appear in the portal. In the past, there were some special other regions that were not removed, but because we want to process everything the same across all GDC in the spirit of harmonization, we're following that same rule with TCGA. So next, I want to talk about whole genome sequencing. For whole genome sequencing, we start out with whole genome sequencing alignments, tumor and normal, and those are compared to generate simple somatic mutation variant calling using Caveman and Pindell, structural variant calling using Brass, and copy number variant calling using ASCAT. And the GENCODE 36 upgrade actually only changed 2 of these files here. There's the structural variation BedP format file that was generated with Brass, and the gene level copy number estimate file that is generated using ASCAT. There were no changes in the alignments, and all of the other variant calling files were also not changed. And this doesn't really, none of these changes really apply to the TCGA whole genome sequencing because there's very few of that on the portal at this time, and they're only aligned reads. So now to talk about some miscellaneous data changes that were done because of GENCODE. The first, I want to talk about copy number variation. These are the ones that were generated using arrays. The gene level copy number files that were generated with ASCAT were replaced with v36 versions. You may remember in the past there were files that were generated from GISTIC. Those have been removed from the portal, and we're going to talk in a little bit about what that means in terms of availability. Um, the last thing I want to mention about copy number is that because the GDC is still determining how to properly visualize data from a different pipeline, the visualization on the exploration portal for copy number variation is temporarily unavailable, but it will return sometime in the future. Lastly, for single-cell RNA-Seq, all of the files have been replaced with v36 version. This includes all of the gene expression, secondary expression, and the alignments. The um, the methylation data, um, in the past we had TCGA methylation liftover data on the portal, and that has since been removed. Um, this liftover data was replaced with our pipeline that we run for the other projects, which right now include HCMI and CPTAC3. This is the Sesame pipeline, and what you will see on our portal right now are the masked methylation arrays in which the germline heavy parts have been removed, and the methylation beta values from Sesame. So this is all available for the TCGA projects now. The Genie liftover files, these have all been replaced with v36 versions. These are all variant files and should all be different now. Um, in addition to all the v36 changes, there were a lot of things that didn't change when v36 happened. So in addition to all the things that I showed you with the previous experimental strategies, microRNA-seq remains unchanged. You should see no changes between the files in data release 31 and data release 32. That's because the microRNA-seq data did not use the GENCODE gene model, but used the mirror-base database for gene location. So that one was unaffected by GENCODE 100%. Non-molecular files, it's basically common sense, but um, clinical biospecimen slide images, because they don't require anything with the gene model, they remain unchanged. The attack-seq data, that was only alignments and was also unaffected by this change. And then the RPPA data, the protein expression data, was also unaffected. So just to give you a little bit of background for how things work in the GDC, is that the GDC, our API has a file versioning system in which new files can replace old ones. If you're wondering if a file is versioned, you can check the version information at the bottom of each file page in the GDC data portal. If only one version exists, this information will not appear. And one thing I do want to let everyone know, because this may affect how you download things, is that if you have one file with a UUID and you version it, you create a new version, the new version will have a completely new UUID. It will never be the same. So here you can see below, this is an example of a version file. Um, versions 1 and 2 are shown here, in which the current version is labeled. And in this case, both of them came out in the same release because they were versioned between releases. But sometimes you will see different release numbers here. And one thing I want to mention, I'm not showing here, and I think this has appeared in previous webinars. It'll probably appear in future ones, but we do have an API that can be used to query versioned files. So if you're looking at large numbers of files and you want to check the version information for them, this can be done programmatically through the API. It doesn't require that you click through all these different file pages. Um, So some pipelines were replaced with others, so they might not appear as new versions of their corresponding file. Um, for example, in TCGA, the ht-seq count files were basically replaced with star counts files, but they aren't actually new versions of each other because they were different pipelines. Some pipelines, like the methylation liftover data, were removed and not replaced at all. These will just simply not appear in the GDC data portal. However, they can be downloaded using the data transfer tool if you do have the UUIDs or an older manifest file. So you won't see them on the portal, but if you have the UUIDs ahead of time, you can still download them, and it should be the same access level as always. So don't worry if you need those files, you can still download them. Um, so now I want to get into downloading GENCODE v36 data and how that can be done. So using the portal at this point, if you just use it normally, you will download v36 data. There's no real trick to it. All of the v36 data is there and available. However, if you did previously use v22 data and you would like to transition to v36 data, we have three options for you. One is that if you use the portal and you remember what your query was, maybe it was something simple, maybe it was all of the lung cancer patients in TCGA, you can still query using the same parameters, and this should yield the v36 version of your data set. So no surprises there. Um, Pretty low-tech way to figure it out. The data transfer tool, the second method, can be used with a special flag called dash dash latest. This will download the latest version of each file, even if you give them the old manifest. So if you have an old manifest using v22 data, you can pass this to the data transfer tool, type dash dash latest, and that will automatically download the newest version, which will be the v36 version. If they don't have a new version, it will just download the same files. The third way that you can download these is that the GDC provides a set of manifests that map the old v22 UUIDs to the new v36 UUIDs. This can be used to download an exact one-to-one set of files. So if for some reason the second method doesn't work for you, these are available if you look in the release notes, and I'm going to link to that at the end of the presentation. And these files, you can use them to map all of your old UUIDs. So generally, the GDC recommends using GENCODE version 36 data because it's more up to date and a lot of them use more modernized pipelines. However, we understand that there are some studies that used v22 data and they're in their later stages. Right now, the papers have been written, the analyses have been done, and you can't afford to go and do every single analysis over again because of a changed GENCODE version. So if it's not reasonable to expect a transition, you can always use an old manifest to download v22 data using the data transfer tool. These old manifests and UUIDs can be provided in a publication to link to the files that were used. Because if you used v22 data, people might want to replicate your experiment and people want to see what you used. So we are making all of the old files available with one important caveat. Because of storage costs and just the sheer size of these files, v22 BAM files that were replaced with v36 may not be available 6 months after the release of DR32, which will be in late September 2022. So we recommend if you If you need v22 BAM files for publication purposes or something else, we recommend downloading them as soon as you can and storing them locally or wherever you can store them. We also have some deprecated files. Like the methylation liftover files or logistic files. These are files that have no v36 version, and therefore they just do not show up in the portal. Um, these can still be downloaded using their original UUID or original manifest. And something that we want to let everyone know is that GENCODE version 22 files will not be generated for data in future releases. All future releases will be GENCODE v36 data, I guess until we use a newer GENCODE version. But for now, all new data will be GENCODE v36. So if you're planning on doing an analysis that includes data that will come out in the future, we very much recommend moving to GENCODE v36. So next we want to talk a little bit about the updates and how this was QC'd and verified. So the GDC bioinformatics team performed a series of comparisons to verify the integrity of the data that was produced with this data model, with this gene model. The results from similar analyses were compared between v22 and v36 data. The comparisons were performed per pipeline on a subset of data, and here I'll introduce a few of the comparisons that were performed. So for the RNA-Seq data analysis, it's expected that the genomic and the chimeric alignments should only have stochastic differences, which was observed, as well as the transcriptome alignment. It should be completely different because the transcriptome depends greatly on the gene model. For the gene expression files, the CpTac3 RNA-Seq star counts were used, using the counts per million normalized, and they found a median correlation of 0.98 in the high expressed genes and 0.95 in the low expressed genes. For the copy number variation data analysis, and this is the copy number variation that came from whole genome sequencing, the comparison was performed for the genes on the major chromosomes, and a very low percentage of the gene sample combinations have different copy numbers between v22 and v36. Over 92% of the gene sample combinations do have the same copy number variant. And the reason that that doesn't add up to 100% is that some genes without the derived copy number, some genes didn't have derived copy numbers because they fall within regions that don't have a copy number segment. And some genes weren't in one gene model or the other. So those fell out of this comparison. And this data source was also the CpTac3 projects using 1,500 samples and the whole genome sequencing CMV files. And I believe that was ASCAD. So lastly, I just want to introduce some ways that you can find more information on GENCODE and what we provide at the GDC. So most of the information that I'm telling you here, and even more things that users need to know, can be found in the GDC documentation website. So this is at docs.gdc.cancer.gov. We have many pages on the bioinformatics pipelines that go in depth for how these were run. And in our data release notes, so make sure when you go to the doc site, you go to the data tab at the top and you go down to the release notes at the bottom of the list there. This gives a full list of the changes that were done for the GENCODE release in bullet points. In addition to that, because these were run using either updated pipelines or slightly different gene models, some aliquots failed the pipelines for one reason or another. And this is all outlined in the release notes, as well as a giant list for all of the aliquots that had some problem that the user should know about. So that's all provided in the data release notes. Um, and then in addition to that, we have the manifest that I mentioned before to transition between V22 and V36. It links to a GitHub page and it's separated out by project. So if you have a specific project you're interested in, you can download that and use it to transition between V22 and V36. Lastly, I want to mention that the GDC Help Desk, if you have any questions about anything that I'm mentioning here or anything that I'm not mentioning here that applies to GDC, feel free to contact us at this address right here. This will go directly to my team, and we do our best to reply within 24 hours and are happy to answer any questions you may have. So thank you all so much, and now we will take any questions that have been typed into the Q&A panel.
Thanks, Bill. There are a couple of questions. The first question is the API going to be, or has it been updated to work based on data release versions? If so, is there a link to the API docs for that?
Yes. So right now, if you use the API, you will see information on V36 files. If you just use it normally, you'll see the V36 files. If you need to see information about what the previous versions were, there's a history endpoint which will give you this information. And you can go to the API documentation by going to this same docs.gdc.cancer.gov, and there's a tab on top that says API, and you can we have a complete thorough guide for how to use just about every aspect of the API there. And if you're interested in a specific thing, I'd recommend we have a nice little search function here that works very well on the documentation site. So if you have some keywords like versions or history endpoints, I'd recommend typing it in there. It'll bring you directly to the page and the section that you would need for the information you're looking for.
Thanks, Bill. The next question is regarding manifest, which maps version 22 UUIDs to version 36 UUIDs. Could this approach be used to identify the GENCODE version associated with specific RNA-Seq bands?
I believe it could. If you're interested in that, what you could use that. However, because yes, that could be used for that purpose. But I would also recommend just going onto the data portal and looking for the files there, because if the RNA-Seq files, if the UUIDs are in the data portal, they are V36. If you find UUIDs that aren't there, then it might be time to consult the manifests, and that will tell you if they are V22.
Okay, the next question, do you have controlled data maps available? I want to discover the new UUID programmatically, but I wasn't able to find, there was an example UUID in TCJ Kirk map on GitHub. Is there a public API endpoint for these maps? Also, I'd rather not scrape the portal file entity page for that file.
Oh, right, yeah. So, yeah, of course you don't want to go through all the portal file entity page. That would be so many requests. So if you have some example UUIDs, there is a way to query the versions endpoint for a specific one. I can't tell you what that query is offhand, but if you contact the support desk, we can definitely help you out with that. So this can be queried programmatically. You won't have to search through thousands of files for UUIDs. Yeah, recommend the history endpoint for the versioning API.
Okay, the next is, can you provide the URL to the manifest files for transitioning version 22 to version 36?
Yes, I'll show you how to get there real quick. Let me go ahead and pause my screen real quick just to make sure I have everything in order. Okay, so if you can see my screen, so we'll go to docs.gdc.cancer.gov and we'll go over here to the data tab and down here to release notes. So for release notes, we want to look at V32. It should be the top one. You can scroll down or we can click on it. And going down to number 9 here, a set of manifests were generated at the project level that map each V22 file to its corresponding V36 file. So you can click here, and this will bring you to a GitHub page that is on our doc site. So right here, we have a list of all of the different projects that are available. These will map. So for example, we can look at OHSU CNL project, and this gives the ID and gives the case ID, aliquot ID, and some other things here. So I think it needs to be looked at raw. So I wouldn't recommend actually using this page here. But this is where the files are located.
Hey, thanks, Bill. There currently there are not any more questions. If anybody has any questions, please go ahead and place them in the Q&A. We'll just give it about a minute or so.
Sounds good.
The next question is where to find information about the version API.
Oh yeah, sure. I can show that now. So let's go back a little bit to the GDC doc site. And if you go to API, so I forget exactly which one it is. I think it's in search and retrieval, but what we can do is we can type in history and we can go to the history endpoint. It's actually under, if you go to, see, it brings you right to the section. It's pretty cool. But it's under search and retrieval here. So you go down to the history endpoint and you can query for any particular UUID using this endpoint, and it'll give you information about the UUID, the version, and more information about its release date, etc. Um, I think there's a few more parameters you could use here, but this is basically how you can use that API.
Okay, thanks, Bill. We'll just give it a little bit more time. And like Bill said, if anybody has any questions about the GENCODE version 36 release after today's webinars, please, contact the help desk. Okay, here's another question. Does the API show previous and following versions for a UUID?
Yes, it will do that. I think the example on the API page is showing a file that doesn't exactly have that because it was generated when we didn't actually have any new versions, when the versioning endpoint just came out. So that's actually something my team should do. The documentation will be updated soon to reflect that. But yeah, if you type in a UUID, it should show you the versions, the previous versions and the current versions.
The next slides and recordings available where? Bill, do you want to show them where things will be placed?
Sure. So going back to the this won't be on the documentation site. This is actually on the regular website. So instead of docs.gdc.cancer.gov, it is gdc.cancer.gov. And you can get to the webinar by going to the support tab and clicking on monthly support webinar. And at the top, of course, you'll see the upcoming webinars, which is today's. And for the past webinars, it takes a little while for us sometimes to go through, and we need to give these proper captions and edit the video. But when that is done, they will appear here. And this is actually it's a YouTube video, so you can watch it on YouTube, and all the captions should be there. And if you want to like search by if you want to search by keyword, you can actually download the captions from YouTube so that you can go to the section of the video that you would that you're most interested in.
Okay, thanks, Bill. Does anybody have any other questions? Okay. Well, if not, we greatly appreciate you attending today's GC webinar, and thanks, Bill, for a great overview.
All
Hey,
right.
everybody have a great rest of the day. Thank you.
Thank you, everyone. Goodbye.